join in a minute?
You can, sir. Speakers.
Yeah, now there is no issue. It got resolved. Thank you.
Speakers. So a very good morning to one and all. So I'm very pleased to invite you all on the Impact Lecture Series, Session 1, which is going to be held online, organized by Nasropat Institute of Pharmaceutical Sciences in association with Institute's Innovation Council. So before starting the program, I'm very pleased and pleased to have a small presentation of our college AV. Thank you. So a very good morning to one and all again. So myself, V. Swati, working as Associate Professor in the Department of Pharmacology, Nasrabad Institute of Pharmaceutical Sciences, feels happy to organize the program and also as a host of the program. So I feel privileged to give a brief introduction for our institution. Nasrabad Institute of Pharmaceutical Sciences is an ISO certified institution that is affiliated to JNTU Kakinada. So our college is approved by AICTE, PCI New Delhi, and very soon we'll get affiliation with NAC accreditation. Our college is established with a vision to be a center for excellence in pharma education, research, training for the rural folks, and to produce human resources of explanation leadership to serve national needs. Apart in establishing global standards of excellence in pharma education, we also focus on many outreach programs to ignite them with knowledge other than academics. So our institution majorly aims to promote and impart the quality education with human values to meet the need in society. So I feel privileged to invite Ms. E. Sweta Prathama Madam, Assistant Professor in the Department of Pharmacy Practice, to give a brief introduction to our beloved Principal, Dr. Jain Suresh Kumar Garu. Thank you.
Good morning, everyone. It gives me great pleasure to introduce our energetic principal, Dr. Jain Suresh Kumar Garu, a leader who knows the way, goes the way, and shows the way. Sir has completed his graduation from Gulbarga University and post-graduation from College of Pharmaceutical Sciences, Manipal, Mangalore University, and PhD from JNTU. Sir has rich experience of 30 years as an academician and administrator. Sir has served as assistant professor, associate professor, and majorly as principal at different colleges in Karnataka and Andhra Pradesh. Apart from academics and administration, Sir worked as vice president of APTI and executive member of IPGA and life member of IPA and IHPA. Sir has 50 paper publications in national and 8 publications in international journals. Sir has attended several seminars throughout the country and organized national level conferences, Epticon 2010 at Hyderabad. Sir had presented 28 posters at national and international seminars and conventions. Sir also had a book publication called Textbook of Forensic Pharmacy. Without any delay, now I would like to welcome our respected principal, Sir, Dr. Jain Suresh Kumar Garu, to give welcome address about today's session. Thank you.
Yeah, good morning. Respected speakers of the day, Dr. A.V. Krishnaraj Garu, Manager R&D, Layla Nutra Pharma, Vijayawada. And another speaker of today is Professor Ragala Ravikant, the startup mentor from Hyderabad. My dear students and registered delegates and the IIC coordinators of Nasrabad Institute of Pharmaceutical Sciences, I'm privileged to invite you all and welcome and thank also you all to this IIC Phase 2 Impact Lecture Series organized by the Institutional Innovation Cell, Nasrabad Institute of Pharmaceutical Sciences. The two topics of today, in accordance with the guidelines of the Institutions Innovation Cell, are aptly selected. The two topics, the first one being pharma innovation and technologies, an overview and all. In that, Mr. Krishnaraj Garu has selected the scholastic skills of students who are actually involved in the research or project works, etc. It is really required and the need of the hour to the student fraternity. The students who are the audience in this particular Zoom meeting is not only the students of B Pharmacy, we have the students of PharmD also, and academicians are there, professors are there from almost throughout the country. We have seen almost crossing 400 registrations for this. I should be thankful to our IIC coordinator and other team of our NIPS for spreading the message towards the folks of the profession. The second one is in the afternoon at 1 o'clock, we have another session on entrepreneurship. Every student who is passing out from a professional college is aspiring to join as an employee itself. Where from these employers have come? These employers are nothing but the entrepreneurs. Every institution and the motivators always tell to the students not to become an employee, but an employer. That's where the seed for the entrepreneurship starts. We wish every student studying in the profession of pharmacy at least try to become an entrepreneur. I think the light will be focused in the afternoon on this particular topic. I take this opportunity to thank Sri A.V. Krishnaraj Garu and also Professor Ragella Ravikant Garu. And I'm sure and assure you that these two seminars will definitely be of much and immense help to the students of all the categories, not only the students, but also for all the delegates. Thank you very much. A special thanks to Dr. Krishnaraj Garu and Ragella Ravikant Garu, and not the least, but the last one is the management of Nasrapeta Institute of Pharmaceutical Sciences, Chairman Sri Koteshwara Garu, MV, and Director Sri Chakri MS Garu, Ramesh Garu, Mittapalli, and also Kishore Babu Garu. Thank you, and all. Thank you very much. Over to Madam.
Thank you very much, sir, for giving a brief introduction to the today's sessions and also inviting our speakers. So now request Dr. B. Satyaprasad, Associate Professor, Department of Analysis, Nasrabad Institute of Pharmaceutical Sciences, to give a brief introduction on our today's resource person, A.V. Krishnaraj Garu. Sir, SP, sir.
Okay, ma'am.
Please join.
Thank you, madam. Very good morning, one and all. It gives me an immense pleasure to introduce eminent personality and the resource person of today's session, Alluri Venkata Krishnaraj sir, to the gathering. Sir is currently working as a Strategic Research Council and General Manager at Laila Nutra R&D. Sir pursued B Pharmacy at Gulbarga University in 1997, M Pharmacy at Andhra University in 1999, and awarded with PhD in Department of Pharmacology from Andhra University. Sir started his journey in 1999 as a head pharmacology department at Laila Group of Companies and working till date in the same company as a general manager, Strategic Research Council from 2016. And sir is a life member of Indian Pharmacological Society and Laboratory Animal Scientists Association. And Venkata Krishnaraju sir acted as an investigator for 30 plus unique patents and 100 plus national place patents. Sir is awarded with prestigious DSIR National Award for in-house research for development of phylloxin in 2007 by Government of India. And sir has a total number of 4 book chapters and attended a total of 35 seminars and conferences. And now I am glad that I got this wonderful opportunity to introduce such a distinguished person to the gathering. And now I request Mr. A.V. Krishnaraj sir to continue the session. Thank you, one and all.
Thank you, sir, for a brief introduction. Sir, Dr. Krishnaraj sir.
Yeah. Thank you for the kind introduction. Good morning, everybody. Yeah, let me share my screen. So I thank management of Narsarapet Institute of Pharmaceutical Sciences and MIC and Institution Innovation Council for giving me opportunity to talk to you. Can you see my screen now?
Yes, sir, your screen is visible, sir.
Right. Yeah, thank you.
Yes, sir.
Good morning, everybody. So the topic I have the topic given by Innovation Council is Pharma R&D, Innovation Technologies and Research. So with relevant to that, I'm presenting good scholarly practices which are essential in drug discovery. Okay, and the main idea of this session is to motivate the students, pharmacy students particularly, and guide them towards research. So the basic objective is to sensitize students and motivate them to pursue innovations, right? So you can see the quote on the right side. In most of the slides, I put these quotes, but I'm not going to read it for you. You can read it for yourself. So yesterday is the past and tomorrow is future, but today is a gift. You should utilize it. So the topics I'm covering in this session is what is the importance of pharmaceutical or life sciences? And what exactly is drug discovery process? How project management can help in drug discovery process? What are the innovative technologies that are helping the drug discovery more efficient? And what kind of skills a pharmacist should acquire if he want to be involved in research activities or participate in drug discovery programs? And what are the expectations of industry? And what kind of opportunities you have in industry? And good scholarly practices. What kind of practices are good for you, for science, progress of science, and also for the society? So you should all aware of these practices and importance of ethics and ethics, particularly in life sciences. So can you hear me well? Am I audible?
Yes, sir.
Thank you. Yeah, basically, I usually don't present in a single flow. I talk and I'll tell you a few stories which can motivate you. And I also ask some questions. Okay, I'm starting with a question. So can you recognize this person? Whoever can recognize, you can text on Zoom or you can respond in audio. Okay, and my next question is, what is most important thing in your life? This also you can answer in the same way. So what do you think? What is most important thing in your life? Different people will have different options, right? So many people consider one person consider one thing as important, some other person can consider something else, right? You just think what is most important in your life. I would say the most important thing in our life is our life, right? Without life, we cannot do anything. So it is most important for us. So keep this in your mind that life is most important and precious. So we can move on to the sessions. Why you guys did join pharmacy? I always ask this question when I meet pharmacy students. Is it by chance or is it by choice? Whatever way you join into pharmacy, you are at a very, very critical place where your skills and knowledge is useful for humanity. Now, I don't know how many of you could figure out who this person is. Now I'm listing his inventions. He's the one who invented ATP and many molecules like aminopterin, hetrazan, which is used till today, particularly for elephant, what we call that, filariasis, and folic acid, methotrexate, tetracyclines. And he is the inventor of first tetracycline. Before his invention, there were no tetracyclines. That means he is an inventor of a new class of antibiotics, which are tetracyclines. And the first molecule he identified is auramycin. And he also identified vitamin B12. So can you imagine so many inventions were done by one scientist? How many of you could figure out who is this person? Yeah, good. I saw someone responded, Pavani, saying that he is Dr. Yellapuragada Subbarao. Thank you. You're right. And he is from Andhra Pradesh and he is from Bhimavaram. Okay. So actually at his very young age, he lost his brothers, two of his brothers, due to some disease, which is called tropical prose. That is like a shock for him. So you can see the quote here, your determination determines your destiny. So this person is an apt example for that, because he determined that he need to find cure for the diseases that killed his brothers. He did it. So now the treatment for tropical prose is use of folic acid and tetracyclines along with vitamin B12. So that means he invented what he wanted to do. So there are many discoveries happened accidentally, but the intentional inventions are very few. So these are some of them. He also invented this molecule ATP. You all know that ATP is very important molecule, which is chemical form of energy in our body. So for everything that happens in our body, ATP is required. He is the first person told that muscles require ATP to function. Okay, but another European scientific group, they said lactic acid is important for muscle function. Later it was proven wrong and they proved that ATP is correct. So Dr. Subbarao's invention is withheld, but later it got approved by European biologists. Initially they stopped. So for that reason, he could not get Nobel Prize. You know that Nobel Prize in Physiology and Medicine is given by the jury of one particular institution in Stockholm. Okay, so all the Nobel Prize winners' pictures are pasted on their wall, right? So he is the only person, they realized that his invention worth Nobel Prize, but they could not give Nobel Prize due to objections from the European biologists. So later they realized that his inventions are right, although they did not give a Nobel Prize, but he put his picture in that same wall. So he is the only scientist whose picture is there on the Nobel wall without having a Nobel Prize. Okay. So determination is very important. You have to set a goal and you have to determine and work hard to reach that. Importance of pharmaceuticals and life sciences. In my initial questions, I asked you what is most important thing in your life. And I also said that life is most important, right? So pharmacy is also dealing with life. So you know what is the role of pharmacy and all. So I'm just leaving it for your reading. But I always say pharmacy is a noble profession. So how many of you agree with me? I guess most of you are agreeing. Can you tell why it is noble profession? Is it because of you, that is pharmacist? Or is it because of the entity you are dealing with, which is drug? Or is it because of the consumer? It is noble profession because of all the three. Unless you have good skills and do right thing, you cannot do justice, because these drugs are like magic bullets. If you give it in a right way to the right person, they can save their lives, right? And similarly, if something wrong happens or with some adverse events, even it is life-threatening, that means same molecule is saving life sometimes and killing life in other times. So for that reason, pharmacist is very, very critical. Okay. And finally, not only the pharmacist and drug, your patient is much more important. Why? Patient is a consumer of your product, right? Can any one of us go to hospital and buy some medicine and come back happily? Rather, if you buy a bike or car, we'll come back very happily, although we spend lakhs of rupees. Whereas if you go to hospital and take a drug from pharmacist, you will not be happy, right? So why you are noble is because of the drug and also your consumer who is a patient. You should do justice to him because he is the most needy person. So out of all consumers, it doesn't matter if you don't buy a car or not, right? But medicine is important for your life or for your healthy life. So for that reason, as the drug is important and patient is important, that's why the pharmacist also become noble because of these two. So giving a drug to the right way and taking care of patients in a very human way. So we are moving on to next topic, which is drug discovery process. How many of you know how these drugs are invented, how much duration it will take, how long it will take, and how expensive it is, and where it starts with? Okay, drug discovery starts with the choice of your disease. Like if you want to invent an anti-inflammatory drug or anti-cancer drug or anti-diabetic, right? First, you have to choose your disease. Second step is your target. So what is target here? So what is target here? So target means, let's say if you are working on inflammation, you want to work on COX-2 inhibition or lipoxygenase inhibition. So you will select particular molecular target to control or mitigate the disease, right? That is called target. So once you identify a disease, then you have to identify the target on which target you can work with. So once you identify the target, then you will try to identify leads that can work on the target. So these leads are nothing but chemical molecules, either derived synthetically or from natural plants or animals, right? These are called leads. So once you test large number of leads against this particular target, you will select potent hits. And once you find potent hits, we call it as leads. And then you will go for lead optimization. In lead optimization, whatever potential hits you got, in that you will go for structure activity relationship studies. That means if you add this particular chemical group, the activity is increased or toxicity is reduced. Like that, you will understand what structures are acting in what way on the selected target. So that way you will try to synthesize or derive a molecule with desired qualities. That means it should give best efficacy and least toxicity. So that is called lead optimization. So once this lead is optimized, you will go with preclinical studies. So preclinical studies we usually do in cellular systems and also in animals, right? Once it passes all these animal studies, these animal studies, how they conduct is based on the regulatory guideline. In India, in USA, they use Red Book as a guidance. In India also, we have some DCGI guidance for animal studies, which is Schedule Y. Okay, these kind of regulatory documents will guide us on what kind of safety studies are to be done in animals. So once these animal studies are done, you can get into clinical trials. So you can see I have listed phase 0 to phase 4. I'm sure that you are aware of phase 1 to phase 4, but phase 0 is a special category that will be given in only cases of unmet needs. That means if there is no other drug for that serious disease and the destiny is only death, if they don't have any other option, then you can initiate your phase 0 clinical study without completion of your preclinical studies, right? So that means this is a special category. Only in some cases, we'll go for phase 0. In other cases, we'll go for phase 1 after preclinical development. So the basic objective of phase 1 is to understand its safety. Phase 1 is given to healthy volunteers. Let's say if your drug is diabetic, you are not going to select diabetic subjects for phase 1. You are going to select only healthy subjects for phase 1, and you will give your drug. You will study how far it is safe. You will check maximum tolerated dose by increasing doses and find at what dose it is causing side effects. Okay, then you will call it as maximum tolerable dose. So this X amount of dose is maximum tolerable for this drug. Then you will get into phase 2. Phase 2, you will do an efficacy study. That means you need to select patients like diabetic subjects you have to take here. And whatever your MTD is there, you will go with very lower compared to that MTD dose. And you will see up to what dose it is effective. Here also you will go for dose escalation, but your objective is efficacy. So now you will see up to at what dose it is giving desired efficacy and at what dose it is giving toxicity concerns. So the ratio between this is used to calculate therapeutic index. If this difference is wide, then your drug is much safer. Okay, once this phase 1 and phase 2 is done, we will go with phase 3 studies. So usually phase 2 studies are done in disease subjects and phase 3 also in disease subjects only. But the difference is with respect to scale. Like in phase 2, we'll work on few hundred subjects, whereas in phase 3, it will run into multi-thousands of subjects and also covers all the ethnic groups where we want to market the drug. That means like Caucasians, Blacks, Mongolians, Indians, or Asians, like that. So it will cover all ethnic groups if you want to take it as a global drug. So that is phase 3 trial. So that also gives you majorly about efficacy and also some details of its adverse events. Then once you move into phase 4, once you complete phase 3, you have to go for filing and get approval from the concerned regulatory authority, either FDA or CDS in case of India. So then once phase 3 is completed, phase 4 will happen post-marketing trial. So why post-marketing trial is required? In phase 3, you have tested everything. Then what's the point of phase 4? So many people will fail to answer this, but the reason is in phase 3, everything is happening in a controlled condition. These are trials that are monitored very carefully by doctors and other experts, right? Whereas in phase 4, the drugs are in the market, patients are using at their homes, right? That means that much control is not there. And this phase 4 gives you information on real-time use. So this is where pharmacovigilance comes into picture, right? These pharmacovigilance reports, whatever side effects that are there for these drugs, when it is used in a real-time scenario in phase 3. Okay, so this is briefly about drug discovery process. And if you see the left panel of this, if you select 5,000 to 10,000 targets, you will end up with 250 leads. Then further, after preclinical, you will end up with 5 molecules that will be entering into clinical trials. And out of this 5, only 1 may reach the destination, that is to the market, right? Now you know how tedious, how resource intensive, and how expensive this drug discovery program is. So you can ask if you have any questions up to this. So shall I move on to next topic? Now, you know that drug discovery program is very tedious and resource intensive. You got to control it more efficiently. Okay. So project management is one way that will make the drug discovery more efficient. Let's say a pharmacist should always learn project management because pharmacy has all the subjects, like you study chemistry, you study biology, you study physical pharmacy, you study pharmaceutics. You study pharmaceutical jurisprudence. That means you are good at regulations. And you also study IPR. That means you are blend of everything. So for a pharmacist, being in project management is very critical for drug discovery process. So what this project management, how it will help? How it will help is that basically we have 5 different steps in project cycle. Project is nothing but a process that has a limited time period, which can use limited amount of resources within limited cost. Right? So this starts with stage 1, that is conception or initiation stage. That means whatever project you want to do, you have to decide what exactly you want to do. In the second step, once yes, you take a call that you want to go ahead with the project, then you have to go for definition and planning. So this is more critical phase. In project definition, you have to write every single aspect. Let's say if you are doing a project in your B Farm or M Farm, so then you have to write everything in one place. Like you need animals, you need some test drugs, you need a room for that, you need some tools to measure certain things. So like that, you have to list everything and make sure that you have everything in place, right? Similarly, for a small project, if you need this kind of information, imagine for drug discovery process, what kind of information is required. So all that should be captured in this phase 2, right? So this is called scope. What all is covered in the project is called scope, and how much cost it will take is budget. So there is a word called triple constraints. Okay, scope, time, and cost. These 3 are called triple constraints. That means you should control these 2. And the central one is quality. You should not lose the quality in this process while controlling these things. Then once these first 2 steps are completed, first 2 steps really take very long time. And when you get into third step, once planning everything is completed, you will get into execution. So while you are executing, you should always monitor the project, and you have to find what are the key performance indicators. So what indicators will tell you that progress is either progressing or delaying, and what indicators will tell you whether you are fulfilling the desired quality or not, right? So all this you have to know and forecast to the senior team, so that if there is any delay or if there is an issue, it can be corrected at an early stage. Then in the third phase is project performance. Sorry. Yeah. Fourth phase is project performance. Here, project manager has to monitor every aspect and take corrective actions to control and make sure that we achieve the quality within these triple constraints. And last one is project closure. Do you think closure need to be a separate step? Yes, it is very much required, and it's also a very, very important step. So why we need this is, although we want to control project scope, timelines, and cost, sometimes it will vary. In Chinese Olympics, what was the cost they proposed? Initially, they proposed that 1.35 billion US dollars will be enough to create all the necessary infrastructure. But it crossed $20 billion. That means from your proposal cost to actual cost, there will be a lot of changes. If you imagine for Olympics, if that is the situation, you can imagine what happens in drug discovery, how tough you have to control, and what kind of learnings you have to take. So like that, then they have to do postmortem. Why this cost so much increased by 20-fold? So this postmortem will help you on understanding what exactly things happened during the project course, and at the end of the projects, what all targets we missed out of the 3. And these learnings will help us in designing next project in a better way. So moving on to next slide, I'm again starting with some more questions. Are we really working hard? How many of us think that we are working very hard and we have a lot of difficulties because of this particular reason I could not achieve this? So how many of us are thinking like that? So what kind of resources we have? We talked about Dr. Ella Pragat Subbarao. So his inventions were in something like late 1800s to 1900. So at that time, what kind of resources they have? They don't have proper electricity. They don't have internet. They don't have several other resources we enjoy, right? And if you go back much more into the history, what are the inventions made by earlier human beings that we are still using? So you can see here, wheel, this was invented in 4th millennium. And fire and controlling fire, it was invented more than 1 million years ago, right? So we are still using wheel and fire for many areas, even today, right? So at that time, what was the situation of human being? Can you imagine? Today, we feel that we don't have money or we have some problem like that. But at that time, if you imagine, to get their food, they have to either hunt or collect some fruits or other food items from the tubers kind of items from the forest. That means just for food, they have to go into the forest and they never know whether they come back or not. That means they're putting their life to just have a food. So we are not having that kind of situation, right? We all are getting good food. We all are having a lot of resources, a lot of funding is there from governments for poor students. So that means in terms of resources, we are way ahead of them and we have a lot of resources. But at that time, they don't have any resources and putting their life, they did inventions like wheel and fire, right? So we should stop giving excuses that because of this, I could not achieve this. Don't give such excuses and put your best of your efforts to achieve it, right? Here, what I want to say is excuses and efforts are two different paths that will lead to either failure or success. You have to choose which path you want to go. So far we discussed that drug discovery has a lot of things to do and it is very resource intensive. It takes a lot of time and it's very expensive, right? So now what kind of innovative technologies that will help us make this drug discovery more efficient? So if you go back, initial days, we used to use only some biochemical assays to understand efficacy of a lead molecule or against selected targets, right? Later, there were inventions. That means in the biochemical experiments, we use just one at a time in a tube, right? But later, when there are micro ELISAs and micro well plates, you are running 96 assays in one go. So that means there is a lot of progress happening. Even ELISA is not that high throughput. So later, there were much more advanced screening systems where large number of ELISA plates will be tested with the use of robotic arms. That means every day you can test 5,000, 10,000 leads. So these kind of technologies are improving the rate of drug discovery. Then if you see multiplex assays, so multiplex assays means in ELISA, you will test only one molecule. Let's say if we are testing T4 or TSH, like thyroid hormones. So you will test only one in one ELISA kit, right? You need to have different kits for different biomarkers. Whereas multiplex assays will utilize very small volume of serum sample or plasma sample, like 5 microliter, and they will test a panel of assays. That means that will contain mixed antibodies, and there are some magnetic ligands to it. That means whatever the small amount of antibody that is there, it can capture particular antigen, like what it happens in ELISA. But multiple such antigens can be detected simultaneously, and they can be separated out because of the magnetic beats, and they can be scanned and tested for their concentration. So that way, using just 5 microliters of serum, you can test 30, 40 different parameters in one go. So that is the advantage of multiplexing. Then if you see, next one is flow cytometry. So far in ELISA or multiplex, you are taking the cell line from the culture and you are freezing that cell line, or you are lysing that cell line and taking the supernatant after centrifugation, right? That means you are taking only cell lysate and testing available proteins in that. So that means you are testing after actually killing the cell. Whereas flow cytometry, it will happen in a dynamic way, like what it happens in our blood circulation. So these cells will be circulating in the equipment, and it will be reacted with certain fluorescent tracers so that you can identify and scan that particular protein. You can see in the middle of this slide, there is an image where black, red, green, and blue colors are there. That means with the tracers, it could separate out different kind of cells. That means it is testing each cell that is exposed to your drug. So that is the advantage of flow cytometry that happens in a real-time scenario. Okay, and you will get the exact difference, how many cells are acting in what way, or how many cells are affected by inflammation or radicals, free radicals test like that. So this will give you real-time information. So moving on, we have more advanced microarrays. Now, a lot of tests can be done using a small chip. If you take DNA microarray as an example, DNA microarray is coded with about 30,000-40,000 different genes. So you can see in the left panel, so this was tested in 2005. So a panel of inflammatory molecules were coded here in this chip. So this will tell, if you add one drug to it, it will tell on what proteins this drug will act. So that means multiple things, not only efficacy, you will also understand the safety. That means if it is suppressing any desired molecule, that will lead to safety issues. So this will help understand your drug much better before getting into actually clinical trials or preclinical trials. This can be done using the cell lysates from cell cultures. So this is one of the very latest techniques which will help in increasing drug discovery. And now more things are coming up, particularly Europe want to get rid of animal experiments. So they prefer cell cultures, but cell cultures are not real models because they don't form in a three-dimensional structure. So later, they invented three-dimensional structures. That means using these cells and using specific cartridges, you can create something close to an organ or a tissue. Okay, so that can also be placed on the chip. You can see the right one. So they made everything into one chip. That means they call this as organism in a chip. That means all important tissues and organs of the body are represented in this chip with the three-dimensional cultures. That means in one go, you will be able to understand what exactly happens in our body. So these are different advantages of new technologies that can improve our drug discovery process. Apart from this, you know that pharmacokinetics are very important for success of a drug, right? Unless it is absorbed and reaches target of action, it's of no use, right? So particularly this bioavailability and tissue distribution studies are very difficult and time consuming. Bioavailability is relatively easy because you can just collect serum samples and get desired data. You will give the drug and you will collect blood samples at different time points, and that plasma you will submit to mass equipments to understand what kind of drug concentrations were reached in blood at different time points. So that will give you about pharmacokinetics of the product. But how you can test whether it reaches to the tissues or not, and whether it is metabolized or not. So this is where it used to take a lot of time earlier. At that time, what they used to do, so they will give the drug, they will collect different tissues, and they will try to isolate the drug-related compounds from the tissues. And that isolates, they used to test chemically and prove that this drug is converted into something else as a different metabolite. So those are very, very tedious experiments. To get tissue distribution data, you need an year or two for one drug. Whereas Now we have new technologies with either radio tracers or fluorescent tracers. If your drug molecule can be attached to this tracer and given through the desired route, either it is oral or intravenous or whatever route it is. If you give and you can scan that organism at different time points, you will know where it reached, in which tissue it is available. So all that advancements are helping new drug discovery to be more efficient. Yeah. Moving on to next slide. Again, we have more questions here. How many of you have your short-term goal? What you want to see yourself, where you want to see yourself in next 5 to 6 years? And what is your long-term goal? What you want to do, where you want to see yourself in 20 years from now? And one more important question I have to you is, have you really learned how to learn? If you know that, if you learn in right way, one topic or one aspect, if you know the trick, then rest of the all knowledge you can acquire very soon. So you should learn how to learn, right? Can you see this picture and tell me who is this person? So this quote suits for this person. Yeah, thank you. Somebody said Ramanujan. You're right, sir. Thank you. Yeah, this person had very limited resources, but he achieved a lot. So one thing you remember that constraints will not stop us from success. Constraints will help us to be more innovative. So these are the inventions of this person, a list of inventions of this person. You can imagine, I think he died about in the age of 30s, 36 or something. You can imagine within that age, he invented so many things. So you should always focus on your goal. You have to set your goals either for short term, that is 5 to 6 years, or long term, that is up to 20 years, or where you want to see best of you in your life. And you should learn the way to learn. Okay. And you should never think that constraints are stopping you from your success. If you have strong desire or if you have strong determination to be successful, then if nobody can stop you, you will be more innovative in spite of a lot of constraints. So now, what a pharmacist should acquire. So far, I told you that life is very important and pharmaceutical sciences is one of the life sciences. And we are dealing with such an entity that is drug that can do extremely good or extremely harm also if we don't give it in right way. Then that our consumer is most needy consumer. So with this kind of situation, what kind of skills pharmacists should acquire? It's very easy to tell that you have to learn all the subjects you have in pharmacy, right? Different teachers are teaching you. They're experts in their own subject. Let's say somebody is expert in pharmacology, somebody in pharmaceutics, but we all want you all to be experts in all the subjects. Is it possible? It is possible if you really understand very basic principles of the subjects. That means whatever small thing you learn, make sure that you learn it completely. So then that will help you learn other aspects very quickly. Okay. And second one is good scholarly practices is very, very important. That means you should know, being a pharmacist or a scientist, how you have to behave, how you have to follow. Okay. So that I'm going to elaborate in next slides. And you should also understand what is IPR and what are regulations. So regulations keep changing, you know, right? Earlier, during World War II, there is no need for teratogenicity studies. Okay, due to certain problem or due to certain disasters. So FDA implemented teratology studies. That means our regulations are changing based on incidents that happens, right? Then integrity, values and ethics are very, very important in life sciences and medicine. As I told you, your consumer, who is the patient, is most needy. So we should be more ethical towards him. And you have to focus on needs of society. Here I have pasted a few pictures. The rightmost one, Dr. Abdul Kalam, although he is in defense research, you know that there is a stunt called Kalam Raju stunt. At those days, for cardiac stunts, they have to depend on foreign countries, and they used to import. They are very, very expensive. So when he started that research, stent cost was 2 lakh. So he prepared a stunt which is affordable for Indians, which is 20,000 rupees. Can you imagine? He reduced the cost by 10 times. That is need of the society. So although he is a defense scientist, so he did that kind of inventions because he has society needs in his mind. Now moving on to next and very important topic for you. So what industry needs, what industry is looking from the pharmacist, what they want, and what kind of opportunities you have. Okay. So industry want a person to have good subject understanding. Why? Means sometimes your project is in different area and industry need in different area. Let's say you did MPharm or any other course of either pharmacology or pharmaceutics or analysis. Whatever you do in your project and whatever you are going to do in your job are going to be different. So you should have, if you have basic subject understanding, you can learn and adapt to the new environment and will be able to excel there. And second thing is updating the knowledge. After learning something, never stop there. You should always follow, once you understand one topic, you should always follow what kind of latest things are happening across the world. One way is reading through such news and also reading relevant journals in the area where you want to become expert, right? Then relevant skills to the position. So if you have the basic knowledge, you will be able to acquire relevant skills that are required to your position very quickly. And next thing is ability to learn. So you should not have any ego and you should not feel that I already completed graduation or post-graduation or PhD and I cannot express that I can't do this. But never say that. If you doesn't know, you can agree that you don't know. That way you can learn quickly. So you should always be open to learn. We all need to learn every day in our life. Then integrity and ethics. As we are dealing with life sciences, it is very, very important. And ultimately, systematic and teamwork. Particularly when you are as a student, your marks or your performances depend on your individual excellency, right? Whereas when you come into industry, you have to work in a team. So this teamwork help divide the task. That means although the task in drug discovery is very big, because of the division among the team, they can do it easily. And that will reap multiple successes. Now, what are the opportunities we have, pharmacists have in the industry? You take drug discovery industry. It's very vast field. Particularly initial phases, you need a lot of knowledge in the biology because you need to select the disease and target. So you need physiologists, pharmacologists here. After that, once you decide on the target, you have to identify leads. That is where you need knowledge in chemistry. After that, you have to test those things. That means you need to have knowledge in analysis and you need to have regulatory knowledge because regulations will keep changing. So like that, in drug discovery, all specializations of pharmacy are required. So everyone will have opportunity in drug discovery, but you need to have and acquire such a skill set that is required by drug discovery. And coming to manufacturing, manufacturing has a lot of opportunities, particularly for pharmaceutics and chemists and analytical also. And to some extent other aspects, but manufacturing, these three opportunities will be more, particularly for formulation manufacturing, as well as the bulk product manufacturing or intermediate manufacturing. So a lot of opportunities are there. And pharmacovigilance and academics, a lot of positions are there as professors and teachers. And similarly, there are a lot of pharmacovigilance jobs. And retail, hospital pharmacy, that I need not tell you. And medical coding and software. Recent days, a lot of opportunities are there in software also. And regulatory and drug control bodies, this is always consistent. And IPR and project management, this is coming up very fast. So why this is important is, let's say IPR, intellectual property rights, this law, whoever did law, they go for IPR law also. Let's say you are a scientist, you have some invention, you go to a lawyer, IPR lawyer. Let's imagine that he is not from science background. You don't know legal language, he doesn't know science. How you will be able to explain your invention to him? So it will be very difficult. So for that reason, if a pharmacist can be part of the IPR, that means if you be in the IPR course and if you learn IPR, pharmacist is more valued IPR than any other subjects, because you have understanding on chemistry, you have understanding on biology, you have understanding on formulations, analysis, multiple aspects, right? So for that reason, any invention that comes to you, you will be able to deal it properly. Same is the case with project management. So for this reason, I suggest some of you should focus on IPR and project management area, where pharmacist's presence makes some difference compared to others. Then this is most essential skill if you want to be innovative or if you want to join in research. I already explained you that you need to acquire a lot of basic skills. One thing is understanding skills. So most of us, what we'll do is we'll satisfy with limited reading. Let's say if you understand definition of something, you feel that I know about that. That is not the case. You need to know what is importance of that definition. So you should try to acquire complete knowledge by understanding it completely. So particularly when I ask pharmacy students, can you explain what is pH? Most of them will answer that it is negative logarithmic concentration of H plus ions, right? They will stop there, but they don't extend to that last word in aqueous solution, because pH has no meaning if it is not in aqueous solution. So that means that is incomplete understanding. Although your definition is correct, it is not complete, particularly for research. Your understanding on small aspects should also be very thorough. You should have complete understanding. Then how you get this complete understanding? Through reading and writing skills. So you have to read a lot. Not only your books or notes given by your teacher, don't limit to that. You have to read relevant journals. So that will help you in two things. One is you will understand how an experiment result is presented by others. So that way, during your project, you will be able to present your experimental results in a better way. Then comes writing skills. If you want to publish, you need to have good writing skills. So these two you have to learn, because once you move into industry, you will not have sufficient time to do all these things, right? So this is golden opportunity. When you are in college or educational institute, you should be able to focus on reading and writing and improve your skills there. Then statistics and design of experiments. Most of us don't like statistics because we don't apply. Once you start apply, then you really enjoy statistics. So without practicing, statistics is very volatile. If you read, you will never be able to retain it for long. Maybe for your exam, it is okay. But always after understanding statistics, you try to take some problems, whether yours or somebody from some other experiment or from literature, and try to do statistics. That way you can retain those skills. So this is very critical in deciding number of subjects to be included in a clinical study and all, and what kind of outcome parameters, how different kind of data should to be dealt, how to deal with normal data, how to deal with skewed data, like that. Lot of information you will have. This will help you. If you understand the statistics at the planning stage itself, you will go for a better design of your project or study. So then design of experiment, this itself is a separate science. I hope there are a lot of books and also information in web. You all should know, all postgraduates in pharmacy should learn these two skills, statistics and design of experiments. So that will help you in your projects. Then IPR knowledge. As I told you, IPR is very important. We also signed that GATT agreement and MAD agreement. So we need to invent and focus on that. Otherwise, we need to pay a lot of money in terms of royalty. So a lot of times, a small inventive step will make a big success. Whenever your time permit, you read how Hoover Dam was built. You can note it, Hoover Dam, which is in California. Yes, it is in California. It is on Hooghly River, I think. So if you see, they could not construct the dam for hundreds of years, but one engineer came up with an innovative tip. You will laugh at that tip. It's very small and very simple, but because of that, they could build the dam. And that is the biggest dam just before Three Gorges. Right? Then you have to present factual outcomes. So most of us like to present favorable outcomes. We always try to suppress or hide negative outcomes from a study. We should never do that. We should always present facts and publish facts. Otherwise, we are not contributing to the science, progress of science. Rather, we are contributing to confusion. So never go for unfactual outcomes. It will be good if your outcome is negative also, that is good. And negative outcomes led to a lot of big inventions. You know how penicillin is invented? You know what is the importance of penicillin? So before invention of penicillin, several people died because of wounds in wars and all. So once penicillin is developed, it saved a lot of lives, right? But you know how penicillin was invented? It's an accidental invention. When Alexander Fleming is working with some bacterial cultures, the bacteria died because of some contamination. So there is some fungal contamination is there in petri dishes. So if he is like us, he would have thrown that petri dishes because it is contaminated. I don't want to deal with it. I'll go for a fresh experiment. But he did it. He started thinking why it is contaminated. Why this contamination killed my cells, my bacterial cells that I plated on the plate. So he thought that way and figured out that this fungus must be releasing something that is harmful to the cells, bacterial cells. So that led to a major invention of penicillin. That's the reason always factual outcomes are important. Don't worry about giving a negative outcome. That's always okay. Then this helps in progress of science. And one more thing you have to keep in your mind is when you design your project or when you design your research, should always focus on your objective first. Why you are doing that? What you want to achieve? What is going to happen at the end of your study? What you can use with that outcome? And is there a utility to that invention? So these are important. So utility to the society and objective of the invention or purpose of the invention is very, very important. So then, as a part of good scholarly practices, you need to learn very basic aspects of every small element. Let's say we are fascinated to see a HPLC instrument or LCMS instrument or NMR like that, right? We all are fascinated with novel approaches and ignore the basics. So for that reason, you always divide your experiment into different unit operations. Let's say you're starting one experiment. What all you need in that? You may need a balance to weigh certain reagents and chemicals, and you need something to measure. You need something to calculate, or you also need to estimate pH. Like that, there are several steps in your project. If you divide them into unit entities, like weighing is one entity, measuring pH is one entity, measuring optical density or absorbance is one entity, like that. If you divide your project into different unit operations and try to learn about each unit operation, you have to see how your balance is telling you how much weight of chemical you are putting on the pan, right? How it tells, So if you understand the basic principle, what is the advantage for you? So if you don't understand, you will not be any different from a grocerman who weigh materials very regularly than us, right? So how we can be different? So if you understand the basic principle in it, the pan on the balance will not be static. It is suspending above the needle, right? So the strain before that will generate electromagnetic force to repel it upwards, whereas gravity tries to pull it down. That means when you turn on the balance, this electromagnetic force try to push the plate up, whereas gravity try to push the plate down. At some point, these two will become equilibrium and there your pan is stable. Otherwise, it is vibrating. You cannot see that with naked eye, but it is vibrating. You cannot start operations on a balance when it is vibrating. Right? If you know this principle, what you have to do is when you turn on the balance, you have to wait for some time because electrical fluctuations will be there. That will cause variation in the electromagnetic force it is generating. So once you have stable electricity and stable electromagnetic field, then you can measure the amount of electromagnetic field. Once it reaches equilibrium, you will tear the balance so that it will consider as zero, right? That means now your pan is in a stable state. That means the vibrations are very small or negligible. So then when you put a load on the balance pan, what happens? You're adding some other quantity. That means gravity will pull down it or push down it further down, right? Whereas magnetic field has to give extra energy to again suspend it. That extra energy is used to calculate the amount of load you put on the pan. So in this process, every step, this kind of vibration will happen. That's why you should not do rush operations. You should wait for a second. And there are a lot of possible errors in weighing. So do you think these errors will really impact your studies? What if you weigh 5 milligram or 5.1 milligram? Is it really different? It is not if you see just as a weight, but you always keep one thing in your mind. What is measurement and what is assessment or estimation? So measurement means something you measure directly. Estimation means using that measured value, you are estimating something else. You are calculating something else. So a small error happens in measurement will lead to a larger error in the estimate. So for that reason, even small errors matter. So you have to understand what are the causes of that errors, like sensitivity error. That means you have to choose right balance. So some people, what they do is if the balance maximum weighing capacity is 10 grams, but they will put 5 grams, then tear it and put another 5 grams on it. So that approach is not right. That leads to sensitivity error and linearity error. So this comes when you are calibrating the balance. And repeatability error, eccentric errors. If you don't place the weight on the middle of the pan, this error will happen. Apart from that, any vibrations in that room or in the table, rush operations or air currents, temperature. So all this matters. If you do everything perfectly, then only your experiment outcome will be accurate. Although this is a small scale, that means you are doing multiples of this. And when you calculate, these errors are getting multiplied. So similarly, spectrophotometer, you use it very frequently, right? Colorimeter or spectrophotometer or plate reader. So you have to understand basic principle of this. I'm sure that you all know it, which is Beers-Lambert's law. Yeah, it's an important question in pharmacy, so I hope all of you know it, right? So in that, there are three terms. One is absorbance, concentration, and path length. So what Beers-Lambert's law says is absorbance is directly proportional to concentration. That is what Beer find out. Later, what Lambert said is absorbance is directly proportional to path length. So when you combine these two, you will derive Beers-Lambert's law. That means totally, if you want to talk about Beers-Lambert's law, it says absorbance is directly proportional to concentration of the solution and path length. When you deal with your spectrophotometer, the width of your cuvette is path length. usual cuvette width is 1 centimeter. That means you unified it, it is 1. So it is easy for your calculations. So now as path length is fixed, based on absorbance, you can estimate concentration. That is how you will be able to estimate a concentration using a standard curve. That means a known concentration of test item, test substance you will take, and you will make a plot, make a graph by testing it at different concentrations, and an unknown substance when you test it, and by extra plotting from the graph, you will be able to know the value, right? So what kind of mistakes that can happen here? So in case of spectrophotometer, the path length is nothing but width of cuvette, right? Whereas in plate reader, the path length is depth of the solution in that well. So instead of, let's say you're measuring, you have some measuring error. Instead of 200 microliter, you put 210 microliter. So this will contribute to two different errors. One is because of the concentration, you have added extra reagent, and also that extra reagent leads to higher path length. The level of solution will be changed. So because of this reason, you should be more sensitive when you are working with a microplate. So that sensitive operation is not required when you are using a cuvette, right? So when you do more number of studies in larger setups, particularly in 96 well plate and all, you should be very careful. If you know this basic principle, you will be able to do that more carefully and more accurately. So always keep in this mind, this may be boring for you, so I want to skip next things a bit. So can you identify who is this scientist? So he is a Nobel Prize winner for thermodynamics. He found the law of thermodynamics, third law of thermodynamics. Now, can you tell what is his name? He is Nernst. So why I took his picture here is today is his birth date. Okay, and Nernst equation is used to derive pH. So this I don't want to elaborate. Like similarly, what I told, importance of basic principles in case of balance and spectrophotometer or plate reader, you have to learn basic principles. A lot of information is there in internet and in your books. But you make sure that you learn. I just want to know, I tell you that how you have to learn, and other things you can learn each aspect like this, what all you are using in your experiments. So now, can you observe this image and tell me what is going wrong here? So there are some issues. Can you notice that? If you look into this critically, you will see multiple issues in this. The levels are not uniform in all the pipette tips, right? That means there is an error. In some tips, you can see something is not dispensed completely. It is attached to the walls. So these kind of incidences leads to measuring error. You know, in a microplate reader, if there is a measuring error, that will lead to multiple error because of change in concentration and change in path length both. So to avoid this kind of situation, what you should do? So there are two important aspects happens while aspirating into micropipette or dispensing into micropipette. Those two are intermolecular forces and intramolecular forces. If intramolecular forces are high, this will not happen. This kind of a small liquid globule attached to that surface and not dispensed. No, this kind of scenario will not happen if intramolecular forces are good. Okay. Then if intermolecular forces are more, intermolecular means the force between these liquid molecules and the plastic molecules in the tip surface. So if that is more, and this kind of scenario will happen, then how you can avoid this? So what you have to do is, when this kind of scenario is there, you have to aspirate very slowly And similarly, you have to dispense very slowly, so that when you dispense or aspirate slowly, this intramolecular forces will pull down the complete liquid. That means the forces between liquid molecules will pull this down and make sure that entire liquid is dispensed properly. So these kind of basic understanding, if you have, you can be a better scientist than the others. Yeah, I think from here, moderator, can you tell me how much time we have?
Sir, you can wind for 5 to 10 minutes, sir, because it's almost
Right,
12.
right, yeah. Okay, so this will not take more than 5 minutes. I'll try to close it fast.
Sure, sir.
So who is this person? Can any one of you identify her? So she is taking presidential award from JF Kennedy. So can you identify this person? So how many of you know thalidomide disaster? One thing I observed in particularly students from Andhra Pradesh is they don't try to express themselves. They don't try to speak out. They have a lot of inhibition that they may be wrong. That's okay. If you express something wrong, immediately others will correct you. If you don't express, our conceptions or misconceptions will not be corrected. Yeah, somebody raised hand. You can answer this question. Mr. Ajay Kumar. Okay, so expression
Thalidomide
is very... Yeah, please go ahead, Ajay.
Yes, sir. It's a stereochemical disaster. I form will give limbs, R form will give normal.
Right,
Sir.
you are right. Thank you.
Thank you, sir.
Yeah. So now, why I took this as example, particularly for Telugu students. Most of us, we are more inhibitive and don't express ourselves. So expressing ourselves is very, very important. That's the reason I'm bringing in story of this person. Okay. So she is the one who averted thalidomide disaster. Thalidomide disaster, I hope many of you know, otherwise you read through. So that led to a lot of changes in drug discovery process. Post to thalidomide disaster, governments and federal agencies, they started new rules like teratogenicity is compulsory for drug discovery, like that. So this is a critical aspect or a big disaster in the history of drug discovery. Okay, but America did not affected by this discovery just because of this person. Okay, so her name is Kelsey. Kelsey received an award from FDA in 2010. Okay, you know the name of award? Name of that award is Kelsey Award. So that means FDA has created an award and named that award on Kelsey and gave first Kelsey Award to Kelsey itself. So this never happened in history. An award has created and given to the same person. Like Alfred Nobel never received Nobel Prize, right? So whereas in Kelsey, she received an award on her name. Now let's look into it. Why she received that? Okay. Kelsey moved to US from Canada after her medical schooling in Canada. So she worked in some universities in pharmacology department for a few years. Later, she got an assignment in FDA. She joined FDA. So she received one file. So she received one file which is asking her to give approval for a drug. So she is the junior most member in the panel. Remaining panel members are very, very seniors. And that company is a big company, and that drug got already approved in Europe and several other countries also. Okay. So that way, this young lady asked one question. You know what that question is? So this drug is for pregnant women to avoid vomiting sensation. Okay. Then her question is, did this drug is tested in pregnant animals? According to animal ethics, you cannot test in pregnant animals. So nobody will test in pregnant animal. And there is no guideline from FDA or any other regulatory body to test in pregnant animal. So this question looks very, very foolish at that time. So remaining members of FDA, they were very mad against her and they forced her to sign that. She said, sorry. Although she is the youngest member, she expressed herself. She said, sorry. Then there is no other way in FDA. So they have to reject that application and asked the company more questions. And they came back with one more filing. Again, she reviewed it and she came up with some more different questions. And it is rejected again. And there is a lot of bureaucratic pressure on FDA because that company is influencing government and pressurizing FDA. But she did not yield it to that pressure. She rejected it thrice. After rejecting it thrice, the company was planning to apply for the fourth time, but they did not apply it because this penguin baby syndrome came out in Europe. So whatever countries, this was used in Australia, a lot of European countries, UK, so many countries, more than 50,000 babies born were without or deformed or very small limbs. So that was the disaster happened. And because of her expression, it saved a country. So Americans were affected, but they were in Europe, whoever are participating in World War II and the army and their families in Europe, they got affected, but whoever are living in America, they did not got affected. That means expression is very, very important. Now, FDA created a prize or an award on name of her. After that, Canada recognized that, oh, I have to do something. She is my citizen. So then they said very late, like after she reached 96, 97 years, they said, we'll give an award to her. At that time, she is so old. She said, I don't want any award. I cannot move from my house. Then the governor or president, they came to her house and presented the award. So that was first time in history in Canadian government. So president or governor coming to an individual and giving the award. So you see, because of her determination and expressive skills, helped her getting this. And moreover, it saved the country from such a big disaster. And now, whoever perform well in FDA, they will be given this Kelsey Award. That means her name will be there forever. With this, I thank you all. So I thank management of Narsarapet Institute of Pharmaceutical Sciences, principal, organizers, and moderator Dr. Swati, and Dr. Suresh Garu, principal, and management of institute, IIC and AICTE for giving me this opportunity to interact with you. So I thank you all for your time and attention. So one thing you please note that you all have great potential. You can do wonders in
Thank you, sir, for a wonderful session. So now the session is open for queries. So anyone who are having any doubts can please raise your hand so that access will be given. So I request all the delegates, if you are having any queries, please, the session is open for queries. Sir, a small doubt, sir, apart from myself. So now present research in silico has been gaining some boom, sir. Sir, is there an advantage of doing in silico research apart with the drug discovery, sir?
Yeah, in silico will be like indicative information, but you have to prove it in proper cell culture or preclinical model. So in silico research is good for virtual things like before synthesizing the molecule itself. You can give your structures to it, possible expected structures, and get the information. Then you have to synthesize those structures. So that can be considered as an indicative data. So that will help you reduce the number of leads required to get some hits.
Yes, sir. In step of lead optimization, in silico is gaining its boom, sir.
Lead optimization, they don't consider to that extent, because lead optimization, you need to have a biological proof. In silico, what happens, whatever information we know about the different targets, different enzymes present in system, that we are feeding in, that information we are giving. And we are also giving drug information, like what is its three-dimensional structure, what kind of forces each molecule have, and all we are giving. That software is matching these two and telling you that these are going to be hits specific to these targets. All this is virtual, and it contains the information we have given, right? So do we know entire biology? So we know a part of it. Inventions are still happening, new things are coming up. So that is not complete, that will not give you a complete picture, but help you to be more efficient.
Lead optimization stuff. So a big thanks from the management and the principal of Nasrabad Institute of Pharmaceutical Sciences, Dr. Jain Suresh Kumar sir, and myself also as an IIC convener and coordinator of this program. So my heartfelt thanks for accepting our proposal and for your well-educated explanation session on research, sir. So our students who are in budding stage, who are showing their steps into the research, your session enlightens us regarding research, sir. So very thankful for you for accepting our session and for this session today, sir. So as a token of our small gratitude, I request to accept a small certificate presentation, sir, in live. Thank you, sir. Thank you so much. So delegates, if you have any more doubts, you can please free to ask. Sir, I have a doubt, question raised in chat box, sir. Shall I ask you, sir?
You please.
Sir, may I know how the drug will be coated to bear metal stents in AP? Any manufacturing companies having that technology? It is from Dr. Kishore Babu, sir.
I didn't get this question.
May I know how the drug will be coated to bear metal stents in AP? Any manufacturing companies having that technology? I mean, coating to bear metal stents, sir. Coating for the drug.
See, actually, to my understanding, nobody is working on coating of stents in India, at least. But some hospitals in Hyderabad, particularly Care Hospital, worked with Abdul Kalam on stunts aspects. Maybe they can approach Care to get some information. I don't have any information in that area.
One more question, sir, from Sri Teja. Design considerations for clinical trials in rare diseases and other diseases. Is there any difference between those?
Yeah, there is a lot of difference. In case of rare diseases, we'll get quick approvals. Before completion of preclinical itself, we can go for FDA. If it is something like small cell lung cancer or any other disease where there is no medicine, they can give you early permission to conduct clinical studies. Like what I mentioned as phase 0.
Okay, sir. So hope Sri Teja ma'am, your doubt has been answered. Thank you, sir. Approximately 50 to 60 students, 60 delegates are in live, and approximately 200 students of our campus have joined in similar hall live presentation. So hope everyone have enjoyed the session, sir. Thank you so much, sir. Okay, sir. Shall I end the session, sir?
Thank you, ma'am. Thank you all. Have a good day. Bye.
Thank you so much, sir. Thank you.